Hi, and welcome. In this video, we're finally introducing two new crosses. These crosses are the start of an attempt to breed my Snow White guppies with my half-black red rose guppy strain. The goal is to transfer some of the red rose vintage to the Snow Whites. We anticipate potential challenges with the Magenta gene, and I'm excited to see the results of this cross. If you're new here, let me quickly bring you up to speed. My name is Ivan, and this channel started as a way for me to document my project of stabilizing a Snow White guppy line. This involved deliberately outcrossing my Snow White male, Gandalf, to non-Snow White females. It's been a long process, and we're nearing the end of this initial phase. I still have a few more crosses to finalize to meet pure breeding standards. However, the plan was always to continue beyond that initial goal, setting new objectives to learn more about guppy genetics and keep improving my lines. Now that the initial stabilization of my Snow Whites is nearing completion, I've decided to focus on incorporating different fin shapes. My personal favorites are the Halfmoon and Hi-Fin Dorsal varieties. While many strains have half-moon tails, particularly dragon guppies, they didn't seem like the right choice for crossing with my Snow Whites. The main reason is that most of these strains have too many colors and patterns on their tails and bodies, which is the opposite of the look I want for my Snow Whites. What I particularly love about my Snow Whites is their uniform, solid color. It's important to me that this characteristic remains even after introducing a new strain. I believe that introducing strains with patterns in their tail or body could lead to unwanted complications later on. So, when searching for a guppy with a half-moon-like tail, my priority was to find one with a relatively solid coloration, which ruled out strains like dragon or even ivory guppies. This is why I ultimately decided on the red rose strain. However, this choice comes with its own set of questions and concerns, which we'll explore by first discussing red rose and half-moon tail types before moving on to selecting our breeding pairs. The major question I have about this red rose strain is whether a rosetail is simply a variation of a half-moon tail, or if they are two distinct traits. It's been surprisingly difficult to find clear information on this, especially regarding the genetics. I haven't found much concrete genetic data on rosetails, and only a limited amount on half-moons. We'll touch more on the genetics later, but just by looking at them, there do seem to be some physical differences. The defining feature of a half-moon tail is its 180-degree spread. To understand this better, let's look at one of my males that doesn't have a half-moon tail. He has what I'd describe as an irregular round tail. Let's zoom in and increase the contrast here. Focusing on the top and bottom edges of the tail, notice how the rays along the edges appear shorter and don't seem to branch out as much as the rays in the middle. To illustrate this more clearly, I found a helpful diagram in a research paper by Lana Reese on caudal fin regrowth in platyfish. While platyfish aren't guppies, this diagram had the clearest labels compared to other guppy illustrations I've found. One interesting thing I learned from this article is that the rays in the caudal fin are made up of cartilage, shown in blue in the diagram, and mineralized bone, shown in red. Reese's article covers more than just regrowth, and I've included a link in the description if you're interested in checking it out. Looking at Reese's figure, you can see those shorter rays along the edges that I pointed out earlier in my guppy. These are labeled as procurrent rays, and they have no branching points. The longer rays in the middle, which branch out, are called principal rays. These branchings are known as ray bifurcations, and they're labeled as primary, secondary, and so on, depending on how many times they split. The key takeaway here is that the procurrent rays on the edges of the tail don't branch out like the principal rays in the center. While it might not be immediately obvious, it does seem consistent in my round-tail male here. I counted approximately 23 rays at the base of the tail. Significant branching or bifurcation doesn't really start until about the 6th ray from the bottom, what's technically called the ventral side. The principal rays show the most branching, with maybe a maximum of 3 bifurcations. Now, let's examine another one of my guppies. This is a typical Snow White male from Cross 11. For this close-up view, I was able to borrow a macro lens from a coworker. It took a little getting used to with its shallow depth of field, but let me tell you, the detail in these close-up shots is amazing. I'm definitely considering investing in a lens like this myself. So if any of you have any suggestions for good macro lenses that won't break the bank, please let me know in the comments. Anyway, let's look at his tail rays. Again, it appears he has around 23 rays in total. Identifying the small procurrent rays along the edges, both on the ventral and dorsal sides, can be quite challenging. In this male, bifurcation seems to start around the 7th or even the 8th ray from the ventral side. And the longest principal rays appear to bifurcate only about 2 times at most. Let's keep these observations in mind as we move on to looking at some half-moon guppies. To get a better look at the 180-degree tail spread, I've gathered some photos and videos I found on platforms like Facebook and Instagram. I'm particularly interested in the caudal fin rays of these guppies. I'll use this bluish guppy as my main example, even though his half-moon shape isn't the most extreme I've seen. However, the resolution of his rays is the clearest among the examples I've found. Zooming in, it looks like this guppy also has around 22 rays at the base of his tail. But, unlike my Snow White male, significant branching seems to start much earlier, perhaps on the 2nd or 3rd ray. These procurrent rays also appear to be longer, which is even more evident in other half-moon photos I've seen. Additionally, the principal rays seem to bifurcate more extensively than in my Snow White male. These observations are really interesting. I believe the half-moon's distinct shape likely comes down to a couple key factors. Firstly, the procurrent rays are not only longer, but they also bifurcate, and this branching helps to spread the tail closer to that 180-degree mark. While that might be enough on its own, I suspect having 4 or more bifurcation points on the longest principal rays also plays a significant role. The sheer number of ray tips likely requires the tail to expand, effectively pushing it out to that full 180-degree spread. So, going back to my initial question, is a rosetail just a type of half-moon, or are they distinct traits? Based on what we've seen so far, a half-moon tail seems to have elongated procurrent rays where bifurcation starts as early as the 2nd or 3rd ray. Additionally, the principal rays should bifurcate at least 4 times, potentially resulting in 8 individual tips per ray. I'm going to use these characteristics as a sort of benchmark to compare my red rose guppies to. Now, let's take a closer look at my red rose guppies. Here are some of the males I have. You can see they have larger and fuller fins compared to regular round-tailed guppies, which becomes especially noticeable when the males flare. The females also have larger tails than my Snow White females, and both sexes sport impressive large dorsal fins, which I really like. While we could discuss the rays in the dorsal fins too, let's stick to the caudal fin for this video. I'm showing you photos of both a male and a female here, because the female's lighter coloration makes the rays a bit easier to see. Looking at their caudal rays, they have a total of around 22 for the male to 24 in the female. The procurrent rays in the female seem to start bifurcating around the 6th ray, while in the male it's a bit earlier, maybe the 4th or 5th ray. These procurrent rays also appear to be about as long as those in the bluish half-moon we examined earlier. So, based on this, my red roses might not be as close to a true half-moon as I initially thought. They definitely show more branching compared to my Snow Whites, which is a positive, and they do have those seemingly longer procurrent rays. However, The key difference is that the procurrent rays don't start bifurcating as early as the second or third ray like in the half-moons I found online. This leads me to believe that my red roses might just be a stepping stone on the path to a half-moon, and I might even need to consider focusing more on just the Hi-Fin Dorsal trait. There's another interesting characteristic of the Rosetail that sets it apart from the half-moon. I've waited until now to show it because it tends to develop in the older males. But here it is. These males can develop additional ray extensions that give parts of their tail a distinctly lobed appearance. I'm quite sure this is where the strain gets its Rosetail name. Personally, I'm not the biggest fan of this trait, as it's not very consistent from one fish to another, nor is it uniform across the edge of a single tail. However, this feature might actually prove helpful in reaching my goal of a Halfmoon Snow White. Let's zoom in on one of my males so you can see what I mean. What I immediately find fascinating here is that the main trunk of the rays doesn't actually line up with the longest points of the branched sections. This seems wildly counterintuitive to me. Let me explain. I initially expected that the ray bifurcations would naturally lead to the longest extensions forming along the same axis as the main ray. Think of it like a tree where the tallest part of the canopy is directly above the trunk. But what we're seeing is clearly the opposite. It's more like a tree that's been pruned into a V or U shape to avoid power lines. The edges where each ray begins seem to form the highest points of these lobes. It's a bit of an unexpected structure. It really appears that these lobes are the result of extensive ray bifurcation, likely even more so than what we see in the half-moons. I'd love to give you an exact count to be more precise, but it's just too difficult to track all these tiny branches. However, it looks like the main trunk of each ray splits into what seems like more than 30 individual rays. Considering that bifurcations typically happen in powers of 2, this suggests that there are probably around 32 individual branches stemming from each main ray, indicating that the rays might have bifurcated approximately 5 times. This is why I'm thinking the Rosetail might actually be beneficial in the long run. A potential blessing in disguise, or at least I hope. My reasoning is that whatever gene or genes are responsible for this extensive bifurcation in the Rosetails could potentially counteract the fin-reducing effects of the Magenta gene in my Snow White Guppies. For those of you who are new to the channel, it's important to understand that my Snow White Guppies need the Magenta gene to have that white coloration. However, the Magenta gene doesn't just affect color, it also unfortunately reduces fin size. This is likely why most show breeders tend to avoid working with Magenta. But lucky for me, I'm not a show breeder, and I want to experiment. One of my key questions regarding Magenta is whether it affects the total number of bifurcations in the tail rays. It might shorten the length of the rays, but does it also mean fewer branches? Or could the same number of branches exist, just at a smaller scale? This is where the Rosetail's high number of rays could be a real advantage. Perhaps they could still push the tail towards that 180-degree line, even with potentially shorter fins due to Magenta. If the Magenta and Rosetail genes can somehow work together in this way, then a short-finned Halfmoon Snow White might actually be achievable. Of course, it's important to remember that mixing genetic traits isn't as straightforward as mixing paint. Often, one trait will simply be dominant and mask others. When it comes to Rosetail specifically, I haven't been able to find much definitive genetic information. My guess is that it involves multiple genes and isn't easily explained by simple dominant or recessive inheritance. However, I have found some information regarding the genetics of Halfmoon and Hi dorsal traits. And this comes from the gene reference table compiled by Alan Bias and Carl Groenewegen. According to Bias and Groenwegen's gene reference table, the halfmoon tail shape appears to be controlled by a sex-linked gene on the X chromosome. They also note that this trait is often observed alongside Hi-Fin dorsals, which aligns with what I'm seeing in my red roses. The term homocercal or homocercal caudal here simply means that the tail is symmetrical on the top and bottom, unlike, for example, the uneven tail of a lower sword guppy. What's also encouraging is that they used a guppy that looks very similar to my rosetails as one of their visual examples of a half moon. This suggests that there might indeed be a genetic connection between my rosetails and half moon trait. The genetic information for the Hi-Fin Dorsal is quite interesting. It seems that the exact way it's inherited isn't fully understood, whether it's dominant, recessive, or something like codominance. However, the table indicates that it's inherited autosomally, meaning it's not linked to the sex chromosomes. It'll be fascinating to see how this trait is passed down in my guppies. So, that wraps up our detailed exploration into the genetics and fin anatomy related to this new crossbreeding project with my Snow Whites. However, I want to be clear that I'm not an expert in this field, and it's certainly possible that I might have misinterpreted some of the information we've discussed. I haven't found a lot of comprehensive data specifically comparing half-moon, rose, and conventional tail types. So at this point, my conclusions are largely based on my own observations. If I come across more definitive information in the future, I'm definitely open to adjusting my approach. Alright, so now it's time to move on to the exciting part. Selecting our breeders and getting these new crosses underway. Because of the possibility of sex-linked genes, I've decided to set up 2 reciprocal crosses instead of just 1. That means we'll be pairing Snow White males with Red Rose females and also Red Rose males with Snow White females. I've already selected 2 Snow White males, and I'll be using 4 of the 5 Snow White females that I showed you in my last Cross 11 update. However, I haven't yet chosen the Red Rose pairs for either cross from my available group. So let's take a look at our potential candidates. I have a good selection of Red Rose males to choose from, but I've decided to avoid the large ones. These tend to be from the earlier broods of the original parents I purchased, and I suspect they might be a bit slower due to their larger fins. I want males that will be active breeders and able to keep up with the females, so I've picked out these 2 younger males that show a lot of potential. They're both vibrant and full of energy, which I'm hoping will translate into them being great breeders. When it came to the females, I looked for those with the strongest caudal fin spread and also the straightest top line. That's the line from the top of their head to their dorsal fin. Unfortunately, the top lines on some of these females aren't the best quality. Some have a bit of a raised area near the head, and others have a top line that almost curves inwards. However, I'm hoping that by outcrossing them with my Snow Whites, we can introduce some new genetic diversity that will help correct these top line issues in their offspring. So, these are the 4 females I've decided to go with. Alright. The last step is selecting the Snow White females. I have 5 available and I've decided to use 4 of them. The reason for this is a bit of redundancy based on what I'm seeing in my Cross 15 project, which will be the focus of the next video. For some reason, the 2 females I selected for that cross keep producing unhealthy fry. Since these 5 females are sisters to the mothers of Cross 15, I'm a little concerned there might be a genetic issue at play. However, I can't be completely sure because another sister that I used in Cross 16 is producing perfectly healthy fry. So to increase my chances of getting good fry in these new crosses, I'm going to use 4 females. The remaining female will be used to repeat the pairing from Cross 15 with Thorn. These guppies come from lines that have been breeding somewhat consistently, so I don't anticipate seeing much variation in the phenotypes of the offspring. I also want to mention that all the females I'm using were separated from males very early on, so they are all virgin, and any fry they produce will be from the specific males I've paired them with. I introduced both of these new crosses on April 3rd. The cross with the Snow White males and the Red Rose females I'm calling Cross 13. And the reciprocal cross with the Red Rose males and the Snow White females is Cross 20. I think I'll hold off on trying to predict exactly what the offspring will look like until the next video focusing on these crosses. However, I'm really curious to hear what your predictions are. We've talked about the relevant Snow White genes in previous videos, and those are still important here. There will be some new genes coming in from the red roses, but they might be similar to what we've seen before. At least that's my initial thought. So, let me know what your predictions are in the comments below, and we can compare them to how these crosses actually turn out in the future. Alright, that's going to be all for this video. My ultimate goal is still to achieve a Halfmoon Snow White, and it looks like we've already encountered our first potential hurdle in deciding whether the Rosetail was the right choice. I'm hoping it'll work to our advantage, but if not, we can always shift our focus to that beautiful Hi-Fin dorsal fin. No matter how this cross turns out, I'm confident we'll learn something valuable. If you're interested in following along with this breeding project, consider subscribing. In the next video, I'll be officially introducing Cross 15, which I started a few months ago. As I mentioned earlier, this cross is between Thorn and some of my Cross 11 females, but for some reason, both females seem to be consistently producing fry that are mostly unhealthy in some way. So stay tuned for that. Now for some quick updates on our other crosses. Cross 16, which is between my C9A male and C11B female, is progressing very well. Some of their older fry are starting to show some nice color, and the C11B female has just dropped her second brood. Cross 17, the pairing of Thorn with his daughters from Cross 14, is also underway, and we're still waiting to see their first batch of fry. I also started Cross 18, which is a pairing between a male from Cross 11 and 3 females from Cross 13. I'm quite excited about this particular cross. One of the females just dropped fry, and therefore I will only focus on her broods. To ensure I have a continuous line of young red roses, I've also started an auxiliary cross, which I'm calling Auxiliary Cross 2. For this, I've used a male with an interesting red streak in his pectoral fin, and I placed him in the tank with my remaining red rose females. If you're interested in seeing the lineage of the Snow White guppies I've used for these pairings with the red roses, you can check out this video right here. Thanks for watching, and I'll see you in the next video.